Okay, let me pull up. Let's give folks a second to. Share my screen. Okay, hi everyone. Welcome to today's webinar with Dr. James Berry. Before we get started, I just wanted to let you know that the learning series is made possible by the Gilbert and Jacqueline Fern Foundation and our industry partners, Mitsubishi Tanabe Pharma and Biogen. If you haven't had a chance, I'd highly encourage you to check out some of our educational webinars. We have a My ALS Decision tool. We have 5 of them so far. So that way you can take a look and see if that information will help you. We also have 13 different guides to talk through different treatment options and, of course, the ALS series. And again, a reminder for folks to consider registering for the National ALS Registry, and you can do that at cdc.gov/ALS. And I wanted to go ahead and introduce Dr. Berry. Dr. James Berry leads the Massachusetts General Hospital Division of ALS and Motor Neuron Disease. He's an active clinician and an ALS researcher with a focus on biomarker development and ALS clinical trial methodology. He oversees a large biorepository collecting, storing, and sharing blood, DNA, and spinal fluid and accompanying patient information, which allows researchers around the globe to conduct critical research, identifying biomarkers and developing novel therapeutics. He also leads the project developing digital endpoints for the ALS trials that will help increase objective data for trials while decreasing the burden on trial participants. Finally, he is deeply committed to developing novel therapeutics by translating discoveries in the lab into clinical trials and leading clinical trials that will transform our approach to care. Dr. Berry also oversees and leads the clinic at the Healy Center for ALS, caring for patients and helping to build and lead multidisciplinary care teams. He helped found the Telemedicine for People Living with ALS program and the ALS House Call program and the Parenting at a Challenging Time program within the clinic. So with that, thank you, Dr. Berry, and I will go ahead and allow you to share your screen.
Perfect. Thanks so much. Thank you for having me. Can you hear me okay?
I can.
Good. And let's see if I get this started. Do you see the screen okay? Yeah. So I'm going to talk today about observational studies and sort of what they accomplish for ALS, and then give a few examples of both the things that we've learned and the kinds of studies that we're running now. And I think this is something we think about maybe less than we do about clinical trials. In a way, I think of observational studies as really laying the groundwork for clinical trials. Just to get our kind of terminology out of the way, or sort of, a baseline here, a clinical trial is a human study that evaluates the effects of an intervention, like a drug or a treatment, on health outcomes. It can be in any disease. In this case, we're talking about ALS. So we're basically, in a clinical trial, testing a drug to see, does it work to slow the progression of the disease or reverse symptoms or treat symptoms. In an observational study, it's also a human study where we're observing and analyzing a disease or a risk factor or a biomarker, which I'll talk about in a few minutes, or a diagnostic test without an intervention. So no drug, but we're trying to understand something about the disease or its treatment. So even though I'm going to spend most of the time talking about observational studies, I want to really begin by talking about what we're trying to do in clinical trials. So in ALS, a clinical trial is really attempting to find a new drug. So imagine it a little bit like trying to fit a key in a lock. And in some ways, that's a simple problem. we have a lock. The more keys we try, the faster we get to the right one. And so one might say, well, all our efforts ought to go into clinical trials, right? I mean, that's the logical thing to do. The faster we try those keys, the faster we get to a treatment. And that certainly has a lot of truth to it. But I would also say, and we should put a lot of effort into trials. I would also say that running trials can be complicated. So it's true that an ALS trial is like finding a key, and at the same time, you're finding it from a large pile of keys, when some of those keys in the pile that should be there haven't yet been invented. And each lock is very complicated, and there's a pile of locks, any of which might be the right lock. And we can't always even tell when a lock is unlocked versus locked. One of those in there is unlocked. And each of the locks can be, in some cases, nearly invisible if we don't have the right tools to sort of see it. So this is complicated. And it does not by any means mean that we shouldn't be doing clinical trials. And what it means is that we need our best keys for the best locks forward. And at the same time, we need to simplify our problem. So we need to understand those locks and identify the locks that are important, visualize them, and be able to tell when a lock is unlocked, and develop keys that are likely to fit important locks. And so without belaboring that analogy or metaphor too much, what I would say is that this is really where observational studies come in healthcare, is to sort of say, we can simplify our clinical trials problem by building the tools that we need to build either along the way or beforehand. I do not think that clinical trials should wait for us to have all the answers from observational trials, but we do have an opportunity to sort of build the bridge better as we walk over it. And I think we're in a good place. And I think there's also a lot more that we can learn along the way by having both observational trials running alongside clinical trials. So observational studies build the tools we need to run these good clinical trials. And those include understanding risk factors like genetics or the environment, a social context that may affect the outcomes of the disease progression models, biomarkers, and that could be imaging, biofluids, electrodiagnostic, and we'll come back to biomarkers in a little more detail. Drug targets, we need to understand, what are we trying to affect? What is the lock we're trying to unlock? Outcome measures, that is, how do we measure whether we're changing the disease or not? And they may be clinical measures or what we call patient-reported outcome measures or digital health technologies. And the other thing is that observational studies generate big data sets that we need for AI. I think increasingly, as we think about using artificial intelligence to give us novel insights into the disease, the main thing that AI needs is lots of data. And we can generate some data through clinical trials, and we can generate additional and even larger data sets through observational studies. And that will catapult us forward as a field to understanding how best to treat ALS in trials. So there are a lot of things that we might try to do in clinical trials and, in observational studies. And as a result, there are subtypes of observational studies. So natural history studies, clinical outcome studies, biomarker studies, digital health technology studies, survey studies, registry studies. I think these are all words that we've heard. They all are slightly different. There's a lot of overlap in that we're trying to understand about the disease. But what our focus is a little bit different. And so just to give you a few kind of examples here, different observational study subtypes address different needs. If we need to understand symptoms or develop what's called a patient-reported outcome, that is sort of a survey, basically, that tells us about the disease, we might need to do a survey study. And it could be at a single time point. Maybe we just deliver a single survey. We can do that on the web. Sometimes we can do it in person on paper and pencil. Sometimes we want to do it over time to see how an outcome measure changes, for example, or how people's perspective about their symptoms change. And that might help guide us to some of the most important symptoms in the disease. For example, fatigue is the second most commonly reported bothersome symptom in ALS after weakness. And that tells us something about the disease, and it also tells us something about how we ought to be thinking about what we measure about the disease. That comes from survey studies. If we need a very sensitive quantitative outcome measure, we're beginning to turn to digital health technologies. That is, anything that can be measured by a digital tool. So a wearable device, a smartphone that has sensors on it, something to measure breathing quality at home. These are all kind of digital health technologies. And we might need to measure that DHT or digital health technology along with our standard measures. So ALS-FRSR and our digital health technology so that we can anchor it to something that we are more familiar with. It might even be that we're testing better outcome measures, but we need some anchor to tell us how the disease is typically. If we understand how the disease affects people, if we need to understand how the disease affects people across a whole population, then we'll do a registry study. And that is really aimed at capturing as close to everybody with the disease as possible. It may be longitudinal. It may not be, maybe a single time point. But this is, when we talk about registries, we're really talking about large populations typically, and really trying to understand broadly how is the disease behaving, where are people with the disease, are there hot spots, things like that. If we want to understand how the disease progresses, then we turn to something called a natural history study, which is really capturing clinical measures longitudinally. This can even be done from electronic medical records by capturing data that's already being collected in the course of clinical care. And it really does tell us a lot about what happens to the disease from sort of the medical perspective. That can tell us about the economics of the disease, which can be really important in getting insurance to pay for new medications. It can tell us about what to expect for how things might change in a clinical trial setting and so forth. And if we need markers to help us diagnose, prognosticate, that is, say how quickly the disease might change, or monitor the disease, then a biomarker study is really our focus, where we would capture clinical measures and biomarker measurements at the same time or longitudinally. Now you can see how many of these overlap a bit, and we're doing all of a little bit of all of them, but it's where is our focus? And in some studies we can hit on many of these different pieces, but in many studies we have to be focused, very focused, to get the information that we need and then get out. Now, I want to start by talking about biomarkers because I think they're really key to making progress in clinical trials. And so they are a big focus of what we do in our observational studies in ALS. And just to sort of demystify it, the biomarker definition, there are lots of definitions. This is I think a broad one, but it's an objectively measured characteristic that indicates health, disease, or response to medication. There are much more nuanced descriptions, but for example, cholesterol is something we measure in the blood, and it can tell us about somebody's risk of having a stroke or a heart attack. Blood sugar is something we measure in the blood, and it can tell us about someone's risk of developing diabetes, or if they've developed diabetes, it can tell us about the status of the control of that diabetes. These biomarkers don't tell us everything about risk. They don't tell us everything about the severity of the disease at present, but they do give us some quantifiable window into the disease. And that's what we need in ALS. And I'll show you one example of where we have that now, and it's a powerful example. So there are lots of different ways we can use biomarkers. One is diagnostic, establish the diagnosis. It would be wonderful to have a diagnostic biomarker in ALS, some blood test we could send, and it was absolutely certain that we had the right diagnosis. I think it would reduce our diagnostic delay substantially. Prognostic means how will the disease progress? And we're beginning to have some prognostic biomarkers. Sort of looking into the future, what can we expect? Fast progression, slow progression. And then finally, and there are many others, but just to sort of keep it simple, finally, disease monitoring. And that is to say, how far along has the disease progressed? And this can be tested multiple times, ideally, so that we could sort of say, it was, at one place early on, and then we tested it again, and it had progressed a certain amount. Having that could help us clinically, yes, but it could also really help us in clinical trials, because what we're trying to do is slow the progression of the disease. And if we have a quantitative biomarker that can show us that, it could be really, really beneficial in clinical trials. So some people call biofluid biomarkers a liquid biopsy. It's sort of a way of looking at a fluid and measuring a protein or some characteristic of it and telling us about the disease. Now, in ALS, the fluid types that, when I say biofluid, the fluid types that we think about are blood, spinal fluid, or also called cerebrospinal fluid, urine in some cases, and even saliva. Blood, remember, has many components. So the liquid component can be called serum or plasma. There are slight differences there. Whole blood is just all of the blood, and it includes red blood cells and white blood cells and the liquid component, platelets. And then peripheral blood mononuclear cells. These are the white blood cells, and they can tell us about immune function. They can also be transformed into stem cells and then into motor neurons in a petri dish so that we can study the motor neurons from the disease just by getting blood. So they have a sort of a particular use. They can be more complicated to obtain and to work with, but they have a really particular use that can be very helpful in studying the disease. I think it's worth spending a moment talking about what we call a lumbar puncture and is commonly known as a spinal tap, which is how we get spinal fluid or cerebrospinal fluid. And this is done by inserting a needle in the lower portion of the back. It's a very safe procedure. We can do it lying down or seated. We do it actually below the level of the spinal cord so that there are nerves sort of floating in the spinal fluid there, but not the spinal cord itself, which ends just above your belly button. And so there isn't sort of some, I think people worry that there's some risk to the spinal cord. There isn't. One of the things that can happen is people can get a headache afterward. And we call that a post-lumbar puncture or post-LP headache. Usually it's better when people are lying down and worse when they're standing up, and it can go on for a number of days. It can also be very mild. Now, we've switched over the last 10 or 15 years to using a special kind of a needle called an atraumatic needle that really dramatically reduces the risk. So we've looked at this and quantified it, and we went from about 30% of people getting a headache down to about 5% of people getting a headache, and those are much more mild. So I think it's really important to know that. The reason that we talk about this is that spinal fluid, or CSF, is a rich source of biomarkers. This is the fluid that surrounds our brain and our spinal cord. And it was the first place that we learned about neurofilament, or NFL. And I'll tell you more about why that's an important biomarker in just a moment. But the levels of neurofilament are much higher in spinal fluid. And we needed that in order to be able to test it at first. And then we could move to blood tests. There are still other molecules, for example, chitinases, which are inflammatory and neurodegeneration molecules that we can test alongside neurofilament that seem to have a meaning for the disease. And there are many others that we're beginning to be able to test there, like cryptic exons and a number of other proteins that may be good biomarkers, and we need to test in cerebrospinal fluid. And then lastly, I'll say in a clinical trial, if we're trying to understand if a drug gets across the blood-brain barrier and sort of gets to the brain and the spinal cord, being able to detect it in the spinal fluid is really, really important. What we don't want to do is, have a whole clinical development program only to learn at the end that the drug was never really getting to the brain or the spinal cord to begin with. So we often have to include these in early clinical development, early clinical trials. And the other thing about spinal fluid is that we can sample it repeatedly. It's safe to do that. And so it allows us to sort of look at these things before and after treatment and get a sense of how biomarkers may be changing. So let's say we wanted to think about a standard biomarker study. What might that look like? Well, typically we would have a screening or baseline visit where we do a consent and then get routine clinical information and then collect our biofluid. So that might include spinal fluid, or in some cases just blood, in some cases even urine. And then we would, if we wanted to look at it just one time, that's enough. And that can be a very useful study. That can be a very useful study. That is how we began to learn about neurofilament. But we may want to follow it over time. And then we have usually fairly simple follow-up visits where we're getting things like ALSFRS-R, maybe vital capacity, maybe some other clinical measures, and collecting our biomarker. And we might continue that follow-up for 6 or 12 or even 24 months, depending on what we need to learn about the sort of the way that that biomarker changes over time or is stable over time. So I've mentioned this a number of times. Neurofilament light is our most important, well-established ALS biomarker, biofluid biomarker. And I think it's important to just understand something about it. Now, this is a scaffolding protein in neurons that gives structural support to the neuron, and particularly to the part of the neuron that is sort of that long projection out to either another neuron or to the muscle. So kind of, we call that the axon. When neurons are injured, neurofilaments, including neurofilament light, spill into the spinal fluid and into the blood. And so you think of this as something that's only inside the cell. If there's injury, it comes out of the cell, and we can test it in biofluids. And what we know is that it is elevated in blood and spinal fluid when there is neuronal injury. Now, you haven't heard the word ALS there. It's not specific to ALS. So there, a head strike or a crush injury to a nerve can cause neurofilament light to go up. But in the context of somebody with ALS, neurofilament light is elevated. And you can see here that it's more elevated than what we might call disease mimics. So other neurologic diseases that might give you progressive weakness don't have the same level of rise in neurofilament that we see in ALS. It's a little tricky because not everybody with ALS has a rise in neurofilament. So we can't use it as sort of a absolute specific or sensitive test, but still it has a very important use. So here's just some data from one of our now, I see that this slide says unpublished. This is not a now published study that looked at neurofilament, and it is increased in CSF. And we also were finding it's increased in plasma, and there's a correlation between CSF and plasma that is very high. And this is really the study, and others like it, that gave us a confidence that we could look in plasma or serum in blood and test neurofilament, and it would really give us similar information to what we're seeing in the spinal fluid. So really, that was really important. And now we're using this in plasma and serum very frequently, still testing it in CSF on occasion, but really relying on it, especially in clinic and in most clinical trials in the plasma. You can see that both in plasma and CSF, as the ALS progression rate goes up, goes faster, you can see that there isn't a rise in neurofilament. Also, it's not perfect. So there are some people who have a slow progression who have more elevation than a few of the people who have a fast progression. So there's kind of a middle ground where it doesn't really distinguish. And that's, I think, to be expected. It's not a perfect marker. The other thing that's interesting is neurofilament is stable over time in ALS. And this is one of the many studies that sort of demonstrate that in plasma and serum and in spinal fluid. So what we see is that neurofilament increases either before the onset of symptoms or early in symptoms, and then really remains stable over most of the course of the disease. That means that if we measure neurofilament level today or in a year, it will give us the same information, which will tell us about, sort of correlate with the progression rate of the disease. But it doesn't go up as the disease becomes more impactful on somebody's life. This is data from Michael Benatar showing that in people who carried a gene that was causative of ALS, we could see the rise in neurofilament just before and just after the onset of those symptoms. And then we begin to see that plateau. So it could be useful, and we use it sometimes during the diagnostic process. Having said that, it is not specific to ALS, and not everybody has a large elevation of neurofilament. And so we have to be cautious in how we think about it. I think really key to our use of neurofilament was the tofersen study. And what we saw there is that neurofilament decreases with successful treatment. So tofersen, which is a treatment for only SOD1 ALS, so this is people who have a mutation in the SOD1 gene and are treated with tofersen. We see that in red is the is the neurofilament level of people who were treated with tofersen. In blue is the neurofilament level of people who were treated with placebo. And you can see a very, very clear reduction of neurofilament of between 50 and 60%. So very robust signal. And then there was that dashed up and down line, vertical line is the time when the placebo participants began getting treated in the open label extension. Whoop. Oh, okay. Sorry, I thought I had an animation here. What we see in the second part of the study is that once those placebo-treated participants begin to be treated with tofersen, that their neurofilament also comes down. Very convincing evidence that tofersen can reduce the neurofilament, and then it slows the progression of disease. So now neurofilament can also be an endpoint for our trial if we think that we have a medication or a study drug that might slow the progression of the disease and protect motor neurons, then we might expect that the neurofilament would come down. We're not quite at a place where we're certain that every study drug that will slow progression will also reduce neurofilament. So we're still sort of wrestling with how much do we rely on it, but we would be very, very excited to see a reduction in neurofilament in clinical trials. And all of the sort of work for understanding this up until the tofersen trial really came from observational studies. And if we didn't have people to participate in those studies and a focus on those studies, we never would have known to put this into trials. So really, I think a success. There are many other fluid biomarkers that we're looking at. I talked about chitinases, MCP1, IL-18. These are all inflammatory markers. We can look at inflammatory markers in peripheral blood mononuclear cells. We have markers of TDP-43 abnormality. So TDP-43 is a protein that becomes sticky and clumped inside of motor neurons in ALS. And that causes changes in other proteins. So we're investigating HDGFL2. There'll be a quiz on that at the end. But this is a protein that changes, is sort of misspliced. It becomes a shortened version of itself when TDP-43 is abnormal. And then we can even look at markers of muscle atrophy, thinning of the muscles. And these are routine clinical tests like troponin T, which we think of as testing in heart attack, but actually can become elevated from ALS itself a little bit, not quite as much. And creatinine, which we test frequently as well. So biofluids represent a really important way forward. I mentioned that AI and computational medicine are another reason to do observational studies. And that's really true. And natural history studies can sort of create these big data sets so that we can use AI. If we want to do a natural history study design, oftentimes it's a little bit of a simpler design in that we'll have our screening and baseline visit, we'll do a consent, and then just gather the clinical data that we need, or maybe just review charts from that point, just review the medical chart. And then follow-up visits may have a very flexible schedule. And the goal is really to maximize the data that we get, but minimize the burden on the participant so that we can, really follow the history of the disease. And these natural history studies have really given us the data that we've needed to do prediction modeling, to sort of get a sense of how do we use baseline characteristics to predict how people's disease will progress. Well, we're at a place where we can do that fairly effectively. It's not so good that we would want to roll this out clinically to say, here's what we expect, but it is good enough that it can actually give us more power during a clinical trial, that is more statistical power, so that we can do smaller trials and still come up with the right conclusion about whether a drug is slowing the disease. So we use these data from observational studies. We do prediction modeling of ALS. We apply that in clinical trials. We update our prediction models and take them back to our observational studies. And that is, that's an ecosystem that really needs to exist in order for this to get better and better. And it relies on us doing observational studies alongside our trials. We can't do this all within one data set, because if you begin to predict, make a prediction on a data set and go back and check it on that same data set, make another prediction on the data set, you actually will just refine your ability to within that data set specifically. And you can get very good in that data set. You take it to another data set, like a different trial, and it doesn't work very well. So you need to go from one study to another, to another, to another. We've also used computational models to really identify distinct ALS-FRSR trajectories. It's not just slow or fast, there are shapes to these trajectories that matter. And using the Answer ALS data set, for example, we've some of the computational biologists that I work with, Ernest Frankel, Divya Ramamurthy, have made these sort of projections about different shapes of decline of the ALS-FRSR, and we're now applying this to other outcome measures to really gain insights into how the disease really does move forward, and you know, that works its way into the modeling that we do for clinical trials as well. And then finally, I think, you know, we think a lot about ALS subgroups, fast, slow, bulbar onset, not bulbar onset. But we can also use AI models and computational models to identify ALS subgroups that may not be quite so obvious. And, you know, it may be a combination of where the disease starts and how the disease progresses and how fast it's going. And these may actually lead us to the biology of the disease better than just our eyes trying to make subgroups of people, which, you know, may or may not be so relevant. But you can see here that we can cluster based on variables that we don't know at the outset, and then we have to go back through and understand what are the kind of things that make these clusters different from one another. We talked a little bit about digital health technologies, and I think, again, I think it's really important to think about digital health technologies in observational studies, because when we want to put these into clinical trials, we really need to know something about the digital health technology. We don't want people collecting a lot of information if that information isn't going to be reliable or isn't going to tell us something about the disease. We want to do some of that work up front before we ever put it into a study, observational or not. And then we want to have our observational data ready so that we can sort of model what will look like. When I say model, predict what the data will look like in a clinical trial, so that we can rely on it. We rely on it for getting a drug approval, rely on it for telling us whether a drug is working or not, rely on it for whether we should move from phase 1 to 2 to 3. And so we need studies that sort of tell us something about these digital health technologies. And, you know, these studies look similar to what you've seen before. We really, instead of focusing on biofluids, we do our consent, obtain our baseline clinical data, and then really sort of establish how we're going to collect our digital health technology for data collection. And then we usually will either collect our data at that visit, or commonly we're using mobile health technologies, and we can send people home for continuous or intermittent use of that digital health technology at home. And there may be follow-up visits as well at regular intervals so that we can troubleshoot the DHT, collect clinical information, and maybe perform some task when people are wearing the DHT to give us insight about how good that DHT is at, say, counting steps, for example. Well, if we want to really know, then we need to have another way of counting steps, and we need to be wearing the DHT, and we can compare to say how good they are. So before I get to some of the kind of wearable technology and mobile digital health, I'll say that we have now for years been studying the ALS-FRSR self-entry, and we started doing that on a smartphone. So this was in a way one of the first digital health technologies that we began to study. And you can see that we had a small study. There was a very high correlation between the self-entry on this of the ALS-FRSR on the smartphone and the clinic-delivered ALS-FRSR. So that they correlated very nicely. And then we replicated this in a clinic-based study and saw, again, a very nice correlation. There were a few outliers here. And when we look back at those, many of those maybe had some change in their cognition as well that you know, may have meant that redirecting during the sort of in-clinic ALS-FRSR might have been beneficial. But by and large, very, very clear correlation here. And there have been some studies now in the US and a very large study in Germany that has shown that the ALS-FRSR self-entry is highly reliable. And so I think this is important as we think about how do we design trials. We've also seen that if we can give the ALS-FRSR more frequently, then we don't have to follow people as long to see changes in it. So again, really might shape the way that we do clinical trials. And digital health technologies also have the potential to really deeply quantify ALS progression and produce these very large data sets that we need for AI. And so we think about things like wearable technologies and apps on smartphones that can tap into the sensors on the smartphones, send all of that data to, secure web servers. Security is, I think, very, very important here. And then they can give us a roadmap. It's not always a very, very simple roadmap. But I think it is very useful. And as we have more and more AI algorithms, we can make sense of data that, years ago we really couldn't make sense of. So one of the digital health technologies that we've been exploring for many, many years is quantitative motor speech. That is just a way of sort of quantifying how people are speaking and using that to quantify disease progression. And one that I've been working on with both Jordan Green and Kate Conahan in the speech lab at MGH, as well as Everything ALS, has been listener effort. And listener effort essentially is answering the question, how hard do you have to listen to understand the person who's speaking? And if people have dysarthria or slurring of their speech, then you have to pay attention a little bit harder to understand the person who's speaking. And if we say, let's use a scale of 0 to 100, imagine that 0 takes no effort. It's somebody who doesn't have any abnormality of their speech and can easily be understood. And 100 would be, you really have to pay attention to understand. Then we can have people rate their own effort in understanding the speaker. And we've chosen to do this with speech and language pathologists who are trained to listen to people with ALS. We've also done it in the past with just everyday listeners, but I think there's a little bit less variability if we use speech and language pathologists. And in our studies with Everything ALS, what we see is that listener effort correlates with the self-reported speech function on the ALS-FRSR. In other words, that question 1 that asks about speech, it does correlate. When people say that their speech is normal, there's very little listener effort. It's a very low listener effort. When people say that their speech is really, really affected, 1 or 2, it takes the listener a lot more effort. And what we're finding is that actually the correlation isn't perfect because the categories on the ALS-FRSR are just too sort of large. And this is a much better measurement. So imagine our new measurement is a real thermometer that goes from -50 up to 200 in degrees. And before that, all we had was somebody going outside and saying it's cold or it's medium or it's hot. The new thermometer is better. And so we have to kind of show that. And what we see in ALS is that listener effort really can tell us about disease progression in a very quantifiable way. In blue, you see controls. There's no change in their listener effort over time. In orange, you see people living with ALS, and you can see that their listener effort goes up over time. And in the background, you can actually see, so you see that bold orange line, which is the group level, but in the background, you can actually see each individual line, and you can see those increasing over time, which is what we would expect. And some are increasing very quickly, and some are increasing very slowly. And that's the variability of this disease. So turning our attention from speech to step counts, we can see that step counts decline over time and correlate with the ALS-FRSR. And this is using a wearable watch called Actigraph. This is commonly used in many medical situations. We also have looked at finger tapping. So we have an app that, I've done this with a few different apps, but we have an app that allows us to sort of count the number of taps that people have. And you can, in a given period of time, so we give the instruction, tap back and forth with your middle finger and your, oh, look at that, your middle finger and your pointer finger. And and we can count those number of taps, and then we can see that that correlates with the fine motor subscale in the ALS-FRS. We've also experimented with using a wrist-worn accelerometer. So this is a watch that's worn on the wrist, and using it to actually look at arm movements over time. So there's a lot of calculation that has to go into this, and a lot of the methods had to be developed really from the ground up, just to look at when people flex their forearm, turn their forearm, extend their forearm, and we can quantify that over time. So in this figure, what we did is take this is showing one person's arm movement. So on day one of the study, this single person, every time they move their arm, we drew a line onto this plot. And you can see that there are many, many lines. And I believe this is the left arm, actually. So you can see that that's kind of the reachable space is sort of, out to the side, kind of away from the body. And we see how many times people do that. This person does that in day one. And then we can look at day 29. And day 57, and day 85, and day 113, and day 141. And we can see that there's a reduction in the number of times that people are moving their arm quantitatively. And this could be a very powerful outcome measure. There are always things that we have to deal with in our data. And one of them is the person wearing this watch for the same amount of time on day 141 as they are on day one? And one of the reasons that this was a powerful one to show is that we do know about wear time. But that's an important thing. And anytime someone shows you data, you need to ask the questions that are basic but important about that data. Was the person using the device the same amount, for example? So we can also use observational studies to inquire about things that are beyond the scope of many of our clinical trials, but may impact our clinical trials and may impact clinical health, even if they're not important in the setting of a clinical trial. And one of those things is social determinants of health. So these are non-medical factors that impact health outcomes. They've been studied very heavily across many areas of medicine. For example, social isolation is associated with a 32% increased risk of stroke. And chronic stress from social isolation appears to increase the risk of seizure in people with epilepsy. So, we have clear ways, and this is just, things that are adjacent in neurology, but these have been studied in heart attack. They've been studied in many, many different areas of disease. I think they've been understudied in the ALS field. And we can modify many, if not all, but many social determinants of health, and they can meet with better health outcomes. And so understanding them could be very, very important. As an example, the previous Surgeon General wrote this book called Together, which outlines really the medical cost of loneliness, and has many, many important facts about how loneliness affects our population and can affect health outcomes. And it may be that people living with ALS are at higher risk of loneliness. And if they are, then we need to know that and understand the impact of that loneliness. And it may be something that we can modify and may have an important impact on both quality of life, but also how quickly the disease progresses. And that's really powerful. Social determinants of health may impact ALS progression. They're widely recognized as medically important, and they really need to be studied in ALS. And they're currently a part of our all ALS study that I'll talk about in just a minute. One of the ways that we might study sort of these social determinants of health is through a survey study, which I mentioned earlier, but really can be very simple. We can do a screening and a survey where we do a consent or even sometimes a waiver of consent if it's a general enough survey. People take the survey, and then there may or may not be follow-up visits at regular intervals, depending on the design of that. So this can be one of our most simple study designs. And again, these social determinants of health are incorporated into all ALS. So I'm going to take just a moment and talk about the all ALS consortium and the studies that are going on there as an example of an observational study in ALS. And what you'll see when I talk about this is that it actually combines a number of our different kinds of observational studies. And part of that is because this is a very large, very complex effort with many, many different investigators involved, many different sites, lots and lots of people with ALS. And I think we have the opportunity to see really important things for our disease and create resources that will drive the field forward. So the structure of this is that there's a project called Accelerating Medicines Partnership in ALS. And this is one of many AMP programs. So there's an AMP for Alzheimer's and an AMP for Parkinson's disease. And now we have an AMP for ALS. And that's powerful because that in and of itself puts ALS on the map with federal funding, as well as with private partnerships that come with that. The All ALS Consortium is a part of that AMP ALS effort. And the goal here is to generate data and samples that can be shared across the ALS community and drive our science forward. There are coordinating centers at Barrow Neurologic Institute and Healy Center, and we share protocols across all of the sites that are participating. We have a shared database and ethics oversight, and all of the biofluids that are obtained go to a central repository called BioSend, and all of the clinical data that's obtained will go to the AMP ALS knowledge portal or ALS knowledge portal and be shared very broadly. So the idea here is really to create an openly used, we still have to make sure that people are doing science, and we still have to have them, have a data use agreement, for example, so that we're not sharing it just so that you go to a website and download it. But as long as it's being used for science, very open sharing of this data. So I mentioned there are 2 coordinating centers. We work very closely together. There are 35 clinical centers around the country. You can see it doesn't really divide perfectly into west and east, but that describes where the coordinating centers are, the east being at MGH and the west being at Barrow Neurologic in Arizona. The goals really are multiple: to accelerate research outcomes by collecting and sharing large amounts of clinical data, as well as some novel patient-reported outcome measures and clinical measures. We want to integrate data, combine data from this study with past studies as well in the single database. We're working very closely, I think I have a slide about this next, with the CDC registry, for example, and Target ALS and all FTD, to really integrate, stitch together that data across these different studies so that people can feel confident that when they're involved in a study, that study is contributing to the broader good of the science, whether it's all ALS or one of these adjacent studies. We're also building community through webinars. We have a webinar later this week, and newsletters and social media, really trying to bring people together so that they know not only about all ALS, but about other studies as well. And we want to improve accessibility. So we have 35 centers across the country, but we also allow for completely remote participation. People don't have to go to a center at all. And that really is different than the way that we ran these studies before, particularly because we're getting biofluids. And so that means that someone will come to do a blood draw in the person's home. So I mentioned this, we've really coordinated with the Natural History Consortium, the CDC National Registry, Target ALS, and all FTD. And we're reaching out to many, many others. We also have worked with ALS TDI to really make sure that the data that we're collecting is not only harmonized and can be used together, but in some cases, creating a clear link between the studies so that we can actually line up the data from one study to another study. There are 2 studies that are actually being run through the All ALS Consortium right now. The first is called Assess All ALS. And this is a study design that's really hitting a lot of the different kinds of observational studies that I talked about, but it's enrolling people living with ALS and controls, that is people who don't have ALS. We're doing consent, getting a fair amount of clinical information, blood, and in some cases, spinal fluid. At home, people do digital surveys monthly on their cell phone and a speech recording. And then we have follow-up visits every 4 months, either in the clinic or at home, where we get these clinical outcome measures as well as blood. And again, in some cases, spinal fluid. And then we have a second study called Prevent All ALS, which is enrolling people who have a are at risk of having a genetic predisposition to developing ALS. They may or may not know at the outset of the study. So they can consent and get all of these clinical, baseline clinical characteristics. There are digital surveys and blood draws that are done quarterly, and we have follow-up visits each year. And because it's so important to tell whether people could be developing symptoms, those visits are done in person. This is actually already quite outdated. This was as of a month ago, and I meant to pull the slide and give updated numbers, but we're really between 375 and 400 participants now, which really speaks to, I think, the excitement of this study. We're enrolling at a pace that just is not seen in ALS studies, and I think the whole community has said that this is an important study to do, and we're trying to do this study well in service of that. So the idea for this really came from the ALS community, people living with the disease, as well as researchers saying we need this information in these samples. So very exciting. It's been really wildly successful, getting a huge amount of data. If anybody would like to connect with All ALS, our website is all-als.org. You can follow that QR code if it shows up on your screen. We're on clinicaltrials.gov, in the Niels Trial Finder, and we have an email as well, info@allals.org. So if you reach out to our study in any of those ways and wanted to be involved, our patient navigators would connect you with the closest site that could do in-person or remote visits, depending on which study you're interested in and how you'd like to participate. So really exciting to have this sort of pulling together like that. I'll stop there. And I can go to some of the questions. I don't know, Lauren, do you want me to read through some of them?
Sure. Do you want me to read them, or do you got them up?
I mean, I have them here, so I can probably read these out. So
Okay.
first of
Sounds
all,
great.
thanks for your attention. It might be just simplest for me to read this out. So when you say that NFL is stable over time, what kind of variation are you allowing for? Is there a certain percentage of natural deviation that's to be expected? How much do we need to look at for a treatment to be effective? And this is a really good question. I'm so glad that this was asked, because there is variation in neurofilament, and there's always variation in a test. There's a small amount of variation in the test for neurofilament. It's actually a very good test, pretty robust. There's also some biological variation, so this number can change over time. We're still working out using large databases of neurofilament and trying to incorporate new databases of neurofilament as they come on. We're still trying to sort of work this out, but we think that there can be as much as a 20 to 30% variability from one time point to another time point. Now, that variability seems to look mostly like this, with people kind of going, up and down. Understanding exactly what that means for individuals over a certain period of time is really important. So let me give you an example. So let's say somebody's going, up, down, up, down, up, down. If you catch them at their first visit on one of their ups, and then you only measure it one more time, they might come down, and you might say, oh, look, their slope is down like this. But you've made the wrong assumption. You actually are just measuring it over too short a period of time. So the other question is, how long a period of time and how many times do we need to measure this so that we can sort of tell what someone's real trajectory is? And I think we probably need to measure it over at least 3 months, but at least 3 months. And we need to measure it, 3 or 4 times, I think, to really get a sense of where it's headed. We also are beginning to say, we probably need a 20, maybe even 30% reduction in neurofilament in a group, in a trial, to sort of say we think that is because of something other than chance. In other words, the drug we're testing seems to be reducing neurofilament. Now, this is all being updated. These are sort of our raw thoughts in the community. A lot of it's been presented. A lot of it is also just conversations in the hallway to try to understand how everybody's thinking about this. It's evolving, and as we get more data from our observational studies and clinical trials, we can kind of update our thinking about that. So small changes may have less importance than bigger changes. And we might even be able to marry that to sort of when do those changes happen and when would we expect them to happen based on a drug treatment. So in tofersen, it took 3 months to see the maximal reduction. So that's important as well. really, really hot topic for us to think about. What's a normal neurofilament number and how much higher is it in ALS? So neurofilament actually varies a little bit by age, goes up with age. And so so it'll be a sort of a cutoff for normal will be a little bit higher in people who are older. There is one confusing thing about neurofilament as we start to use it clinically, especially. Different labs have different normals, a lot different normals. So they can be off by 6 or 7 times from one lab to another lab. So you kind of can't go back and forth between labs. And so it also makes it a little bit hard to say what's normal. There'll be a normal range listed for whatever lab it is that you get. And ALS, people with ALS on average, have higher neurofilaments. Most people do have a higher neurofilament, but there are a few people who are slow progressors who can have a neurofilament level that falls within normal. That's uncommon, but it can happen, and we have to have our eyes out for it. So I think it's we're sort of understanding, how we think about those levels as well. But important to know, if you go from one lab to another lab, you can't just sort of compare those numbers.
Dr. Berry,
What do we know about?
are general neurologists ordering NFL, or that hasn't entered their radar screen yet to help with the diagnostic delay?
Yeah, I mean, I don't know that I can make a generalized statement about that right now. But I would say that I think there's probably some uncertainty still amongst most general neurologists and certainly primary care physicians to order neurofilament. I think it's still being used primarily in ALS centers. That may change. There are things that make neurofilament go up that are not ALS as well. And so I think, we want to be cautious and optimistic about its use by neurologists and even in primary care settings. So I think there's reason to be really thoughtful about that and how it can help us, and also thoughtful about how we want to, roll that out without too much certainty, I guess.
Makes sense. Thank you.
But I also will say there are lots of tests that we have out there that are not perfect. That sort of, we have to interpret the numbers in the setting of the clinical presentation, the person's symptoms and what might be going on. So this is not unique to this test in any way. We have lots of tests like this. There's a question about plateaus. And I have a little bit of an eye on time. So what do we know about plateaus? One thing I can say is that they're we can pick out slower progressors and faster progressors in the data. I think we need more attention to how do we define a plateau quantitatively. How long does a plateau need to be a plateau in order to call it a plateau? So I think when we develop things like the knowledge portal, the ALS knowledge portal, that will be a very powerful resource for us to then say, okay, here's how we define a plateau. Now let's go to that data and find out how frequently we see that kind of a plateau. So naively, I thought I would be able to answer that question long ago by looking at data sets, and it's a complicated question to answer. Just finished the Actimyo study using a wearable device on leg and wrist for a year and taking a survey once a week. Are you familiar with this? I know I won't get personal results, but man, it'd be nice to get something back from it. So, it's all for the greater good, but selfishly, I'd like to know. Yeah, so I'm one of the investigators on the Actimyo study, and it is important to say that it's actually going on not only through Mass General, but also we have a site in Egypt and one in Belgium. And it builds on technology that has been actually approved as an endpoint for another disease called Duchenne muscular dystrophy. So this is a good sensor, and I think a good way of analyzing the data. And I think it will be very exciting to see. We, in fact, are hoping to have a poster at NCALS, which is the European Network for ALS research, sort of equivalent to NEALS. And what would be really nice is actually to be able to sort of present that poster then to people who who've been in the study or even just the public. So maybe we'll try to think about doing a webinar to sort of show what we're learning from that study. And thank you for that idea. And Katie Burke, who's involved in this study, is here today in clinic. I'm in clinic, so I'll talk with Katie about that. I like that idea. In Assess All ALS, what determines whether you take CSF and not just blood? So there are the main question is, what is the participant willing to do? And some people are willing to provide CSF and others not. There are some sites that are focused on CSF for all ALS, and there are some sites that are focusing on CSF for the target ALS study. And in order to knit these studies together and make sure that we're not sort of asking the same person to do the same thing twice, the sites that are focusing on target ALS will first offer somebody to be in target ALS if they're interested in providing CSF, and that CSF will be shared with the community, and it would be a wonderful study to be involved with. If they filled their quota in target ALS, then they could potentially offer it through all ALS. Sites that are not a part of the target ALS study would offer it through all ALS. So it's mainly determined by whether a person is willing to give spinal fluid, and it's very valuable for the field. Thank you.
Well, I think
There's one thank you, which is I really appreciate.
I think we're at time, and I just want to say thank you so much, Dr. Berry, for joining us today. And this will be up on our website if folks want to view it again or had to hop off early. Again, extremely grateful to you and all of your colleagues for really helping define what this looks like and explaining a little bit more for our community. It's been extremely helpful. And next April, and next month actually, we are going to have Dr. Goutman give a talk on ALS exomes for us. So perfect timing with talking about the environment for April. So thank you so much, Dr. Berry, and we look forward to seeing more and learning about all these wonderful projects that you and your team are helping to spearhead.
Thanks so much. Really, thank you for having me. This is wonderful. And boy, Stephen Goutman will give a wonderful talk next month, I'm sure. Yeah. So look out for that one. Thanks again for having me.